who I had that was actually meeting after us talking for a very long time, we just see each other 2 dimensions, which at least for me is 1 dimension more than personalities. But fun fact about Zoe is that she was the New York City inner tube water polo champion.
That's correct.
Which, to me, kind of anything. That's it.
All right, we're done. That's it. So Morgan, you guys are so prompt. I'm incredibly impressed. Just a quick reminder, if you have note cards left from the breakout session, if you can sort of get them to Ruchira or Morgan, that would be great.
Not blank ones.
Yes, only with writing, preferably in English. Okay, so I just wanted to begin. So we're going to transition from sort of whole organism manipulations to how you can manipulate gene expression up or down in specific brain regions, potentially in specific cell types. But I do want to point out that even though I'm the one giving this talk, it's probably not fair that I'm doing so because the person doing all the tool development and all the work is my postdoc, Meredith Lowe. She's here. Find her, talk to her. We're happy to help you out. Yep, right there, where that finger's pointing. Okay. So one of the reasons why CRISPR has been so appealing is that very broadly, we went from being able to manipulate specific sites in the genome taking months and months and months to weeks to now a process that you can perform in days. And a process that's highly tractable across organisms. So Dev highlighted this in that all you really need to target a location in the genome is your guide RNA. Which means you don't even need the sequence of the entire genome, you just need the sequence of your gene of interest. So the take-home point from my talk is that we can help you to either up or down regulate your gene of interest in a specific brain region. And I put this up here, the central dogma, just to remind you that where we are right now is we're trying to control transcription. So we're trying to either block transcription or upregulate, activate transcription. Okay, so why would we even want to do this? The pictures of the whole organism knockout that you guys saw from the octoptocin receptor, they're beautiful, right? But it turns out that sometimes you don't want to knock out a gene in the entire organism, and part of the reason why you might not want to do that is because of pleiotropy. And so I'm just going to give you one example from the vasopressin system. So we know, the field knows, that vasopressin coordinates many different aspects of social behavior. So it coordinates partner preference behaviors, parental care, especially paternal care, selective aggression, and space use in the wild. Now, the thing is, if you knock out the gene, you're going to potentially impair all of these phenotypes, which is useful, but it doesn't tell you much about the underlying neurobiology. What parts of the brain are actually involved in this? And there's sort of a plethora of work in this area that suggests that different brain regions, vasopressin receptors in different brain regions, are responsible for mediating these different aspects of behavior. And so just as an example here, there's really nice work from Miranda Lim out of Larry Young's lab suggesting that vasopressin receptor in the ventral pallidum is important for partner preference formation. There's work from Su Shin on the role of the lateral septum in paternal care. There's very nice work from Brennan Aragona, Kyle Gabroki, and colleagues on the role of vasopressin receptor in the anterior hypothalamus and selective aggression. And then from the Phelps lab, Miriam's work showing that differences in vasopressin receptor in the retrosplenial cortex are linked to differences in space use and fidelity in these guys. So this system, what I like about this is if you think about it, it gives you one gene that coordinates many behaviors where from an evolutionary perspective, you could tweak one receptor population within the brain without affecting this suite of behaviors otherwise. So you could say, lead to differences in paternal care without affecting preference, space use, selective aggression. But in order to parse that out, what we need is a tool that enables us to change gene expression for specific genes in specific brain regions. Okay, so how can we do this? This is not the first attempt to do this, even in prairie voles. So there's some really nice examples out there. We basically covered this why you wouldn't want to knock out. Oh, okay. So you wouldn't necessarily want to do this in the whole organism because you knock it out everywhere for the entire lifespan of the animal. You might also be interested in development. Receptors can often have different actions on behavior at different sensitive periods of development. Okay, so if we want to do this, then we need a tool that allows us to go in and manipulate a specific gene. And that tool that we're going to get to is CRISPR, but how do we get it into the system in the first place? Now this is a phage, so admittedly totally not the right virus, but it's so much prettier and it's good for explaining the point here, which is that at the end of the day, all viruses are is a delivery system for genetic material. And so we've been able to, we meaning scientists, have been able to take advantage of this by taking our genetic material of interest, synthesizing it or cloning it, and then essentially putting it into the virus and letting the virus do the hard part for us by putting that genetic material into the cell. And so the way that we use this typically in the laboratory is that we take our viral prep and we go ahead and we inject it into a specific brain region of interest. Now your brain turns out really dense, which means that you have a limited spread of the virus, making it optimal for targeting specific regions within the brain. And so this is how we can deliver a genetic tool into a very specific part of the brain. Now, in terms of manipulating gene expression using viral vectors, this has actually been going on for a fair amount of time in the Vole world. So we have classical examples of overexpression using AAVs. So here's some, again, that paper from Roland Lim showing that overexpression of vasopressin receptor in the ventral pallidum is sufficient to endow meadow-voles, our promiscuous species, with the ability to form a partner preference. There's also been attempts to try to reduce gene expression. So this is an example from Katie Barrett's work showing that she can actually knock down oxytocin receptor, in this case with RNAi. In addition to this, you could imagine, based on the talk that you heard from Dave, that it would be very sort of straightforward to take the tools that he's using in the embryo, package them into a virus, and just start knocking out genes in specific brain regions using those tools. And that is actually underway, but is not underway from my group. So Arjen Bender from Larry Young's group has a poster here, and so please go find him if you have a question about how to knock out or use the nuclease activity of Cas9 in order to disrupt genes in specific brain regions. All of these have been incredibly fruitful approaches, but all of them have limitations if you're looking for a tool that sort of does everything. And so this is just showing you, what are some of the things that these tools can and cannot do, and what are some of the considerations with respect to their use. And so what I'm going to tell you today is that there is actually a set of tools that we're developing in which we can meet every single one of these criteria. So we don't have to mutate the genome, we can multiplex it at the same or multiple genes, we can manipulate individual transcripts or splice variants, we can use the same system to upregulate or downregulate a gene, we can titrate the system so that we can massively upregulate or just sort of more physiologically relevantly upregulate a gene or downregulate. The nice thing is you're working with the endogenous locus in the genome itself. So you're not trying to package a big gene into a tiny virus. And it's potentially reversible using some of the same approaches that we have in mice, such as doxycycline-inducible systems. Okay, so Dave introduced this idea that what Cas9 does is it uses a guide RNA to find a location within the genome, and then it just chops the DNA. But a group sort of CRISPR 2.0 asked the question, what if you just get rid of the nuclease activity? What does that leave you with? So if you get rid of the nuclease activity, then what you have is a homing system for the genome. So here you still have your guide RNA, which is shown in blue, which is providing the specificity. It's being scaffolded by Cas9, but now when it gets dragged to the genome, it doesn't cut the genome. So this creates almost unlimited opportunities to drag any tool you want to a specific location in the genome. I'm not going to belabor the plethora of tools that now exist, but simply to say that you could take this complex and you could add to the Cas9, you could fuse it with something like a transcriptional activator or a transcriptional inhibitor. So now you're driving your transcriptional activator to a specific location in the genome, enabling you to essentially turn on a gene. Okay. So when we started this project, we were actually considering whether or not we should do sort of the nuclease-driven knockout approach, or if we should go after this CRISPR, what's called AI, or activational inhibitional approach. And right at that time, there was this nice paper that came out of Jeremy Day's group at UAB, where they had put forth a neuron-optimized version of CRISPR activation, which is virally driven, so it can be delivered into the brain. And so we teamed up with Jeremy Day's group, and we are now implementing these systems in Voles. Okay, so here's the system. Basically what I showed you before in another schematic here, we have our DCAS9. DCAS just means dead. So catalytically dead DCAS9. here with the guide RNA in orange loaded into it, fused to this transcriptional activator. So when this lands in the genome, it drives transcription of whatever is close by. Now, the system that we're developing is essentially a mix and match system, meaning that you need to package not only your guide RNA in one virus, but your dCas9 VPR in another virus. Now, this is not required. You could package both in one virus. The reason that we're pursuing it this way is that it allows you to have a large batch of this virus that you can mix and match with as many guide RNAs as you want to try in your system. Okay, so some notes about viruses. So viruses differ in a lot of ways. I'm going to go through two of the most commonly used. So adeno-associated viruses and lentiviruses are both good for delivering genes into neural tissue. And they differ in a couple of key ways. So AAVs are preferred often because they're BSL-1, which basically means no biological safety threat. And they have a larger in vivo spread, largely because they're tiny and they can actually get through that dense tissue of the brain more easily. But the problem is that you can only put a little bit of DNA into them compared to lentiviruses. Lentiviruses, because they integrate into the genome, do require a higher level of biosafety, but something that can be accommodated by the majority of research universities. They have somewhat smaller spread when they're injected because the virus is bigger, but they can accommodate over twice as much DNA. So when we're talking about our system, basically you can put your guide RNA into either of these because it's nice and small. But if you want to use this sort of big, huge, bulky dCas9 VPR activator, this only fits within a lentivirus. Okay, so if you wanted to do this in your own lab, what would it look like? Well, the first thing you would need to do is design your guide RNA of interest. We haven't talked about this at length, but this is something that's pretty easy to just have a conversation with one of our working group on how we do this. There's also a lot of blogs out there that will help you. You then take your guide RNA sequence, you clone it into your viral backbone. Then you're also going to make lentiviruses from this plasmid, and you're going to need essentially 3 lentiviruses. So you're going to need your activational complex that incorporates your guide RNA, but you're going to need to also express your guide RNA, and then you're going to need a control. Then you're going to take your system and you're going to inject it into your Vole, Vole, whatever brain you'd like to do. And then you need to figure out whether or not your system is working. So this can seem a little bit overwhelming, but one of the things I'm here to tell you today is that you can actually outsource this entire top line here. So, and also, I see people taking pictures, and that's great, but we are going to put these talks on the website, so feel free to come back to it later. I can relive the pain forever. So we can help you with the guide RNA design. You can order your guide RNA, and you can send it to Vigene or any number of other companies that do cloning and packaging, and they will clone it for you, and then they will package all 3 constructs, and it's about $5,000. But we are working to reduce that price by essentially creating repositories where you don't have to buy giant batches of virus, but can buy more like 100 microliters at a time. Yeah, well.
Yeah. How much quantity does that give you?
So this is going to give you 500 microliters of each of these viruses, which, depending on how much you wind up diluting your virus before you inject it, is enough to last you like a decade or more. So if it works, so the risk here is if one of these doesn't work very well, you've just spent $1,500 packaging a virus that's now sitting in your freezer making you sad. But if it does work, you have enough that once you've validated it, you're going to have plenty for multiple experiments. Okay, so you can outsource this, or we can help you with it. So we've been optimizing sort of very, very straightforward cloning strategies. We can teach you how to do this if you want, but you're by no means required to do so. It's pretty easy to outsource. So all of that said, the thing that you cannot outsource is injecting this thing into brains and figuring out whether it's working. So we can help you with stereotactic injection if that's not something that your lab currently does. But once you have it, then you're sort of in this territory of figuring out whether or not it worked. And so just to go back to the beginning, we're back here at the central dogma. We want to see ideally that our tool, which is acting at the level of transcription from the DNA, is actually leading to a repression, not only repression or upregulation of your RNA, as well as your protein. And so there are a variety of ways to do this. My lab can help you implement quantitative PCR for measuring mRNA levels, and we can also help you with in situ hybridization. We've been using the RNAscope, and just in case you aren't convinced, here's some beautiful pictures from my postdoc and from Cassie, who spoke up earlier, who's an undergraduate here at UT Austin, who spent the summer in my laboratory, we can not only detect specific transcripts, but we can detect at this point up to 3 at a time. So we can look at co-localization as well. So we're happy to help you. But also RNAscope has great resources. You don't actually need us for this. You can call them, they'll send someone to your lab because it's expensive and that's how they make money, is by convincing you the system works. Okay. We can also work with you at the protein level. These techniques, I think, are a bit more standard within the field, so we can help you with Western blots, autoradiography, immunohistochemistry, which is spelled incorrectly. And so ideally, looking at these multiple levels. Okay, I just want to mention that most of the talk is focused on the activator. That technology is a little bit better developed, but we also have the same thing for CRISPR-I, meaning inhibition. And so here it's actually fused to the crab domain in MECP2. No one knows why it works better if it's actually got that MECP2 on there, but it does. And so in summary, we're here to help you up or down regulate your gene of interest in a specific brain region. At this point, we're just looking at neurons. So we're essentially using the synapsin-driven promoter. There are plans in the work to try to develop more cell type specific approaches. We're happy to talk about ideas that we have in terms of how to do that. But as a first pass, we know that the synapsin promoter works well in leading to expression in Vole neurons. Okay, so how do you get this training? As I mentioned, we do have funds available, so if you want to come to one of our laboratories, you can go ahead and go to the Vole genetic engineering website and fill out this little form, and we'll figure out where to send you and when. And then finally, I want to thank my lab, who are tireless and amazing, and if you come do some training with us, you'll get to meet all of these wonderful people. And with that, I'd be happy to take any questions with the caveat that I might look at Meredith occasionally and ask her. Okay, got it.
This might be a bit of a naive question, but how long-lasting is the CRISPR upregulation or downregulation? How quickly does it take to work?
So we haven't done a time course yet with the lentiviruses. With AAVs, we see sort of anywhere from 5 days to 2 weeks, depending upon the serotype. Serotype, you're going to learn a lot about when Steve gets to talk in a few minutes. It's essentially just the packaging of the virus and how it recognizes a cell and gets into it. In terms of the long-lasting nature for both lentiviruses and AAVs, you have very, very long-lasting expression. So for AAVs, we've gone out to more than 4 months. For lentis, when you use them to put them in the genome, we know we can go out a couple generations, but they do get shut down. You're never going to go out a couple generations here because you're not actually changing the germline, you're just changing the somatic cells. Cool. So very long lasting. Okay, Dave says lenti in the brain, 3 to 4 months as well. Cool. And I'm pretty confident it goes longer than that, just that nobody wants to pay the per diems to find it out.
I think it would be also useful to include HSVs, because sometimes you're going to see changing behavior, then back to baseline.
Can you repeat that?
I think it would be useful to the field also to include HSVs.
HSVs, yes.
Yeah.
Okay, so just for background, that's herpes simplex virus, and HSVs are really useful because they lead to transient expression, so on the order of about 12 days of expression. And so that's one system, and it also has very large packaging capacity. So that is a system we're not currently exploring, but I agree it will be useful if somebody wants to take that on. We're willing to help. That enables you to go in and say, reduce gene expression for 10 days during development, during a sensitive period.
With the activators, do you think that you get expression in cells that don't normally express it? And then, what is the advantage of that over just using the old virus with the gene in it?
Yes. So one big advantage over using the virus with the gene in it is if you have large transcripts. That's not typically a problem for, say, oxytocin receptor or vasopressin receptor, but there are quite a few transcripts out there that are just too big to package into a virus. So that's one instance in which you're not going to be able to use viral overexpression. And then in terms of the cell type specificity, there are some, so with respect to, I'm going to look at you here, but the VPR can only activate in acetylated domains. Is that right?
So it's a little bit more plastic dependent. So Depending upon the transactivation domain, you can actually have certain CRISPR activators that only work in cells where you have open chromatin. And so theoretically, if there was open chromatin for a locus in a cell that doesn't normally express it, you could force expression where it normally isn't. And then there are some actually, depending upon how big that scaffold is, that can actually force expression in a region or a cell type that normally doesn't express that because it's compacted. So you can manipulate it in multiple ways depending on what you package.
But you can bias the system towards cells that are already expressing the transcript.
I'll just talk over. At least in mice, at least in mice, there now are a couple of papers that have come out using the simplest viruses that basically you can put in the brain. You can force expression of a gene only in the neurons that actually normally express it, just at higher levels. So it is pretty restricted using some of these tools.
More training questions?
Don't forget, you can write them down and even ask them later. Okay,